Okay, today we're going to continue forward with some cell cycle information, but specifically now we are going to look at mitosis in all of its minutia and glory. And so by the end of this, the objective is for you to understand each of the phases of mitosis and so that you can identify their names as well as the major events and occurrences that occur. So in order to do that, you need to be able to answer our essential question for the day. What are the steps and events in each stage of mitosis? So there's two reasons that cells divide instead of grow. If a cell grows too large, it has trouble moving enough nutrients and wastes across the cell membrane. So a growing cell needs more food and it makes more waste, and it just gets to the point where it cannot, through transport means, move those things across the cell membrane at an efficient enough rate to keep the cell alive. The second reason that this occurs is that the larger a cell becomes, the more demands that are put on its DNA. And so we must remember that DNA has its limits. So when a cell grows too big, we talked about it has trouble moving the nutrients and the things that it needs. The problem with this is as a cell grows, the volume of the cell increases much more rapidly than the surface area of the cell membrane. And so then when that surface area is not adequate enough to transport the large quantities of things across, that's when a cell dies because it's gotten too large. So all of the information that a cell needs to function is going to be stored in the DNA of the cell. And when a cell becomes larger, there's too much demand put on its DNA to adequately meet the needs of the cells. And so as the cell grows too large, there's this information crisis. And since the cell can't, or the DNA can't keep up with the demands of running the larger cell, the cell dies. So we know that cells have to divide and they don't just grow. But we're going to take a little pause in this information because we need to also consider chromosomes at this point because of that issue of DNA. So in order for the cell to divide, it has to have two complete sets of chromosomes, one set for each of the forming cells. And so it's important that we keep track of what we're talking about when we're talking about this DNA, and when we're talking about them, we're going to be using terms like chromatid and chromosomes. And so before the S phase, there's one chromatid and one chromosome. But after the S phase, there are two chromatids and one chromosome. So humans have 46 chromosomes. How many chromatid before S phase? 46. How many chromatid after the S phase? 92. And so why does the chromatin have to be replicated? Well, the chromatin has to be replicated so that we can actually have two complete sets of information in our cells. So when we're counting chromosomes, it's incredibly important that we remember this red dot here, and it's called a centromere, and the word is right here. The centromere, it is always there. If it's an unduplicated chromosome, the centromere just sits on the chromatid. If it is a duplicated chromosome, the centromere holds the two duplicated chromatin together. So the thing is, when we're counting chromosomes, we always count the centromeres. So we can see here we have duplicated chromosomes. So because we have two chromatid, they make the X shape. But if I count my centromeres, I've got 1, 2, 3, 4. And so I have 4 chromosomes, 8 chromatin. And so we can see that process here. It's probably very important that you go ahead and write that in your notes and draw some pictures because you are going to be required to do this for the rest of the year. So if we think back to our cell cycle information, we had this as our model for the cell cycle. And remember that after telophase and mitosis, we had cytokinesis. I also gave you the acronym IPMAT plus C to remember all of the events of the cell cycle in order. And so we're still working in the cell cycle, except now we're looking only at the M phase. So we've already considered and learned about interphase, but now we're in the M phase, mitosis, and we're going to get some clarity on each of those events. But before we move on, remember the steps of the cell cycle are IPMAP plus C, or interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. You could also remember this as I play music at the concert. Whatever works for you, you just need to know each of these events in order and what actually happens. So we're going to do a fairly in-depth look at what happens in each of the phases of mitosis here. I would have you not write everything down at this point, but just listen, because then we're going to go through and just get the most important information written down for each of these different parts. So remember that interphase and cytokinesis are not part of mitosis, but there are events in both of those stages that have to occur in order for mitosis to be successful. So if we look first, we have to start with interphase. And so when we're looking at cells, cells in interphase are going to have a well-defined nucleus. Outside the nucleus, there's only going to be two centrioles. And remember, those centrioles in diagrams are those marshmallow-looking things. And they're important because their function is to organize the microtubules into a spindle. And as we'll see, the spindle gets to be very important for mitosis to occur correctly. So then we move out of interphase into M phase by starting with the first phase of mitosis, which is prophase. In prophase, the nuclear envelope fragments and the microtubules invade the nuclear area. The spindle is completely formed. The spindle is a structure that will help to separate the chromosomes. And during prophase, the pairs of chromatids become attached to the fibers of the spindle. So we get to actually see the chromosomes during prophase. Metaphase then, our centrioles are now at the opposite sides of the cell. The spindle fibers then push and pull the chromosomes and line them up in the center of the cell. And each chromosome then is going to be connected to a spindle fiber at its centromere. Once everything is lined up in the middle, the cell progresses into anaphase. In anaphase, the centromeres divide and the chromatids move to opposite sides of the cell. So once the centromeres have been pulled apart and there's a complete set of chromatin on each side of the cells, the cell moves into telophase, where a nuclear membrane begins to form around both sets of genetics. There's a nucleolus in both of those newly formed nuclei, and the cell begins to pinch. This becomes what we refer to in animals as the cleavage furrow. Mitosis then is done at the end of telophase once all of those structures are in place. However, the cell is not completely divided into two daughter cells until the process of cytokinesis occurs, and that's where each cell then pops apart into two completely different cells. However, the cells are identical in every way. So that's a whole lot of information. Let's look at it in its simplest components. So remember, we start with I, interphase, which is not part of mitosis, but it has to occur in order for mitosis to be successful. Interphase includes G1, S, and G2 phases. This is where the cell grows and prepares for mitosis, and the cell copies its DNA. Now we get into the first phase of mitosis, which is prophase. And in prophase, the cell is going to prepare and pair. And so the nucleus breaks down, DNA coils into chromosomes, and spindle fibers form. That's all part of the prepare portion of prophase. Homologous chromosomes then pair up, and homologous means the same. And so remember, all of our chromosomes have been duplicated, and so they line up in their little matching pairs, and those pairs are what's going to be separated during the process of mitosis. And so that's the pair portion of prophase. Once all of that work is complete, the cell moves into metaphase, which I remember as middle, because the chromosomes are all lined up along the equator of the cell, or the center of the cell. The spindle is fully formed. It's attached to each of those chromosomes, and it's ready to move into anaphase, which means apart. Spindle fibers are going to then pull the chromosomes apart toward opposite ends of the cell. And it does this, or the spindle fibers do this, by shrinking in size. The tension then causes the centromeres to release, and each side of the cell gets a complete set of genetic information. Once that's occurred, we move into telophase, or two, and it doesn't mean two cells. In telophase, it means two nuclei, and so you can see it looks like a shelled peanut here. It's still a single cell. However, there's a nucleus on the left and the right sides and centrioles on both sides. That's because there's two of those. The spindle fibers disappear, the new nuclei forms, each nuclei gets a nucleolus. And so then once this occurs, in order for the two daughter cells to be completely formed, the final step of cytokinesis has to occur. And cytokinesis is where we completely split into two identical daughter cells. The cytoplasm has been divided between the two cells. Each has its own nucleus, nucleolus, centrioles, and all of its own set of organelles. So at this point, each of these daughter cells move into the G1 portion of the cell cycle. So what are the products and the importance of mitosis? Well, mitosis produces identical daughter cells. Mitosis occurs in all cells of the body except gametes. And gametes are going to be your sex cells, sperm, and eggs. Those are used for reproduction, and they have a special process that they go through in order to be replicated. But mitosis then ensures that the new cells are able to carry on the same function as the mother cell. Thus, this comes back to that whole important part of replacement, repair, growth, and then asexual types of reproduction. So let's name the phases without looking in your notes. If we look at this first one, the cell is growing and developing, and its DNA doubles. We know that that is interphase. Then we have visible chromosomes, no nucleolus, and no nuclear membrane. That makes it prophase. Our chromosomes are aligned across the middle of the cell, metaphase. Our chromosomes have been split in half to each side of the cell. That makes that anaphase. We have two nuclei. So now we're in telophase, and now we have two completely identical but separate cells. That makes it cytokinesis. So it's important to be able to identify the phases of the cells in actual cells. And so here's a drawing of something more similar to what you're going to see under the microscope. And so looking at this, we can see here's a cell that's in interphase, just living its life. Here's a cell that's in prophase. We can start to see that spindly chromatin showing up and condensing into chromosomes. Here's metaphase, when everything is aligned along the middle. We have anaphase, where it has been pulled apart. Here's telophase, because we can see two nuclei in a single cell. And there's cytokinesis, where we have two identical cells formed. Watch these simulations by opening your link to your notes. We are going to watch this one right now, just so you can see what it looks like as a cell goes through mitosis. You'll see up here in the image, we have an actual video of a cell going through mitosis, and here is the animation of what is occurring. What I want you to notice is what is happening and how each phase just flows into the next. There are no start and stop points. So remember that your cells are going through this process about every 24 hours. And so it is an extremely important and fast process. So how long does this process take? As we said, 24 hours. Some are a little faster, some are a little slower, but on average 18 to 24 hours in order for a cell to complete that cell cycle. So go ahead and summarize your notes on this topic by answering your essential question in the summary section. Also, once you're done, go ahead and answer these questions on the Check Your Learning portion of your workbook. And thank you.